Hey, so I guess listening to a talk on biology at a Web3 conference is very, very unorthodox. But just rest assured, I'm also a tech person. My background is definitely cryptography and stuff like that. So I'll try to keep it a minimum. However, that said, there is one picture I wanted to share. You've probably seen it in middle school or high school. It's essentially cell division. And the really neat part about this is that it happens with almost every organism on this planet. It happens with almost every one of us, that whenever a cell divides, whenever we grow, whenever we reproduce, essentially we start out with one single cell, and that single cell duplicates its internal mechanisms, its internal machinery, and then just splits into two. And one cycle later, it splits into 4, 8, and you have an exponential growth. And essentially, a human baby, when it's born, it's made up of about 3 trillion cells, but it all started out with one single cell. And that has some very, very profound implications, specifically that even though it started out as that one single cell, that cell had to contain the entire blueprint for that human body. And the implication is that if we can somehow access that blueprint, if we can somehow understand, decipher that blueprint, we have or hope that we will have the capacity to solve all genetic, all inheritable diseases. Now, if we want to do that, we need 2 steps. One of them is we need to somehow get that information out of the cell biologically. And the second is we actually have to interpret it somehow. And the first part seems very, very hard, and it's actually solved. So the first human genome was sequenced in 2003. It took 13 years, about $2.7 billion investment. However, ever since then, sequencing costs really plummeted. So currently, if you want to go to, if you go to a lab, if you would like to have your genome sequenced, it costs approximately $400. Their actual material cost is about $100. So from a practical perspective, sequencing is solved. Now, of course, we're at a privacy event, and most people here probably are not really comfortable with just going to any random lab to have their genome sequenced. For those people, the solution is not ideal. You can actually do it at home. It requires weeks of preparation and learning about it, and about $20,000 worth of equipment. Now, we can say that, okay, $20,000, it means it's not solved. But compared to $2.7 billion, it's a fraction. And if you are willing to wait 5 more years, the trajectory is going down. So even though it's not necessarily solved, it will be solved. So it's on a good trajectory. However, so I mentioned there that this is the hard part because this is the physical part. Once you actually can take your genome out and sequence it, then the only thing you need to do, it is very, very simple, just decode it, just interpret it. That's the easy part. And the easy part looks like that. Ever since DNA was discovered, we have an exponential growth in research publications. These are the papers that contain the word DNA published on PubMed. So essentially, we have an explosion of data available. Now, one problem is I selected one specific paper. It was written by one of my friends. You don't necessarily have to read it or understand the title, but what I wanted to get at is that even though we have an explosion of information being published about DNA, it is so immensely deep and so requires very expert knowledge that there are barely a handful of people in the world who can actually do it. So my friend has been studying genomics for like 15 years. There aren't many people in the world who can invest 15 years of their time to understand it. And this leads us into a very, very interesting problem. If you would like to have your genome analyzed, if you would like to interpret it, if you would like to know what your genome, what kind of secrets it holds, currently the solution is, well, you just go to your geneticist and ask them. Okay, you ask your geneticists. Now, guess how many papers out of the 50,000 that was published last year they actually read? Probably 5 at best. So there's no single person to ask. And what they will do is they will just go to a company and just buy a genetic software that can analyze your genome and just give you back results. But still, you still have the same problem. Let's say you find a small company. A small company that does genomics will have 20 doctors on board. A big company will have a 100, and if you go to one of the big ones like Novartis, Roche, they will maybe have thousands of geneticists on board. But the moment the only 15 people in the world can understand one specific paper, what's the probability that they have a person on board that can understand every single thing that was ever published by genomics and on genomics? And my guess would be that it's kind of zero. You have no chance of finding anybody in any company in the world that can meaningfully analyze your genome from A to Z, everything. And this is an interesting problem. And the thing is, we've been here before, specifically in the world of software. Not so long ago, if you bought an IBM mainframe, then all your software kind of came from IBM. Or if you bought an Apple Macintosh, then Apple gave you all the software. If you bought a Windows PC, then Microsoft wrote you all the software. And the thinking in the world was that, well, they know what software you need. They shipped all the word processing. They shipped all the spreadsheet. They shipped everything you needed to do your work. And retrospectively, that sounds very, very funny when you think about it, when you think from the lens of the open source movement of where we are today, that at some point, everything was developed by one company and we thought it's enough. Obviously, it was not enough. And it's the same thing with genomics. Currently, everything is developed by one lab or a few labs, and the general thinking is that this is enough. And obviously, it is not enough. And the question is, can we do somehow better? Can we somehow scale this? And my proposition is that what if we could recreate the world of open source, this open source movement, but for genomics? Now, if you want to understand it, however, I think we need to kind of unpack. It's very hard to say what exactly open source means. And for me, at least, I try to identify kind of three pillars which are important for me. One of them is sovereignty. So up until the point when every software that you ran was on somebody else's machine, on some university lab, nobody really experimented. So the whole experimentation with software, with open computing, started when you could actually buy your hardware and you could actually YOLO. You could just tear it apart. You can do whatever you want with it. That's when people started to actually tinker with it. Now, that still was not enough. When kind of the normal pieces appeared, Windows, Apple, etc., what they realized is that, well, if they want other people to write software on it, they have to open up. They have to create a platform where all of a sudden it's not just about you can run some program, but it's about you can extend it. They give you pieces of puzzle on which you can build. That was the second very, very important thing. And of course, the third one, people are vain creatures. If we make something cool, we want to share it. So the whole open source movement kind of became possible when we had the internet, when we had the Bitbucket, the whole sharing site, etc., everything. When we could simply send our stuff to somebody else to run, when somebody else could actually build on our stuff. That's when the open source movement really started to happen. And the question is, what stops this thing from happening for genomics? Because, open source works, so why doesn't it just work for genomics? And the answer is, in my opinion, is that we have a few wrinkles which we didn't quite get right. One of them is mistake irreversibility. So with software, if you mess up, you start over. If you lose your credit card, you get a new one issued. If you lose your private key, hopefully you can roll it. With genomics, if you lose your DNA, it's gone. I mean, nobody's getting back. Once it's leaked out, it's out. So it's very, very irreversible. The second is the target audience. With open source software, we built the platforms for ourselves, essentially nerds building for nerds. Now, in the case of genomics, we have a split. Nerds can build a platform, except we are completely clueless about genomics. And my friend can dissect an eyeball, but he's absolutely clueless, or he doesn't actually care about building a platform. And you have this huge gap, which you somehow need to cross. And lastly, this is not necessarily specific to genomics, but even though the internet allows us to transfer data, to transfer our results, to share in that we can just send each other our results, but sharing it and being able to say that, oh, that's cool, and being able to react, it's a social thing. And that was kind of, for example, in the current world, GitHub is one of those places where we kind of associate with open source movements. We say that, well, open source, yeah, it existed before GitHub, but kind of after GitHub, it took a completely different turn. And then the question that I was trying to answer here is that, what can we do? How can we recreate the open source movement, but for genomics? How can we somehow iron out those wrinkles and make this whole thing possible? And for that, just attacking each of these features independently. So the reason open source, in my opinion, the open source movement became possible is because we became able to tinker with our data, to tinker with our machines. Now, if you want to tinker with our genome, then we have to make mistakes affordable. We have to make them cheap. We have to somehow make certain mistakes impossible so that if we just screw around, we don't have irreversible consequences. And for that, one option is that, yes, you can definitely run stuff on your laptop, run stuff on your phone, that's fine, if you are good enough. If you are not good enough, then my proposition is similar to hardware wallet in the crypto world, where you have a specialized device that has certain security guarantees, that has certain protections in place, which makes it extremely hard to misuse, extremely hard to leak your data out. Because once you have one of those devices that you can control very, very closely, then it doesn't matter what you do on it. The device should protect you from messing up. And that's kind of the important thing, to have sovereignty and tinkering capability. Now, let's say you can do whatever you want with your data. The second problem is extensibility. We are software developers. I can write a library. There are a few genomics libraries out there, but they are not built for biologists. So biologists, as I said, they care about biological concept. They care about an eyeball. They care about different cells. They don't care about compression algorithms. They don't care about cryptography. They don't care about any of these things. So what is very, very important is we want to be able to define APIs that are extremely high level, that are targeted for biologists, but at the same time be able to offer the same protective mechanisms. Again, the idea is that it's very, very easy to just create something that's very powerful, but if you're going to lose your data with it, then nobody's going to use it. Whereas if you can achieve the same security restrictions while providing very high-level APIs, then you have a chance of convincing biologists to go wild. And that's, for example, just one of the tests that I ran on my genome. It's a cute test whether it can tell you whether you like cilantro or you hate cilantro. There's actually a genetic marker which can tell you that part. And here the only thing that's kind of important that I want to get across is that if you have a device or your laptop that can do all the data processing in advance and all the data indexing in advance, then you can have very, very powerful applications. And if you can actually sandbox the whole thing and prove that nobody can ever run off with the data, then you can play, or you can actually have an AI go wild and play, which is an important thing in the current world. Lastly, shareability is one thing that's currently missing is there's no place to be able to share genomic apps. So if I make something, it's mine. If my biologist friend makes something, it's his. There's basically no way to cross it over. If you can define a platform which has secure execution, sandbox execution, secure data storage, everything, and you can create a little platform on top, kind of maybe similar to the Ethereum platform, or maybe similar to the App Store, you get two very, very interesting things. One of them is you get the simplicity of anybody being able to publish these apps, so they can just go wild and publish it. And you also have the extensibility, the composability that open source gave us, where one genomic app can actually reuse the results of another genomic app. And this will potentially lead into these biologists not having to reinvent the world, not having to constantly recreate everything. And essentially these three steps, these three paradigms are what what the whole dark bio project is building out. It's, for the most part, functional. You can actually find me to have a demo if you want to. And for more information, we also have a white paper published. So that was a very, very quick roundup. Thank you very much. And find me outside for more infos.